Can gene therapy and CRISPR cure LGMD? Imagine a future where genetic diseases like limb-girdle muscular dystrophy could be fixed with a simple edit to the DNA. While that sounds promising, the reality is more complex. Limb-girdle muscular dystrophy is a group of rare inherited disorders that cause progressive weakness mainly around the shoulders and hips. There are many types, each caused by different genetic mutations. Currently, no cure exists for any form of LGMD. Treatments focus on managing symptoms and helping people maintain their quality of life. Gene therapy and CRISPR, gene editing are two exciting areas of research that could change this in the future. Gene therapy involves introducing a healthy copy of a gene into a person's cells to replace or compensate for a faulty one. For example, in some LGMD subtypes, scientists are working on delivering correct versions of genes like DYSF or CAPN3. In laboratory studies, especially in animal models and cells from patients, gene therapy has shown potential to restore muscle function and reduce degeneration. But these approaches are still experimental and have not been approved for widespread use in humans. CRISPR-Cas9 is a powerful gene editing tool that allows precise modifications to DNA. Researchers have used CRISPR to fix mutations in cells from patients with LGMD. For instance, they have successfully repaired mutations in the DYSF gene responsible for LGMD2B, restoring the production of functional dysferlin protein, which helps muscles repair. Similar work has been done for other subtypes, like LGMD2A, where CRISPR was used to correct mutations in the CAPN3 gene. While these laboratory successes are encouraging, many hurdles remain before these therapies could become real treatments. Most research so far has been in cells or animals, not in humans. Delivering gene therapy or CRISPR tools safely to all affected muscles in the human body is very challenging because muscles are spread throughout the body. Current methods might only target a few muscles at a time. We also do not yet know how safe or effective these treatments will be long-term in people. It's also important to remember that LGMD is caused by many different gene mutations. Because of this, a therapy that works for one subtype might not work for another. Personalized treatments tailored to each genetic mutation will likely be necessary. Even if these therapies prove successful in trials, they will probably be subtype-specific and not a one-size-fits-all cure. For those living with muscular dystrophy, it's natural to feel hopeful about these advances. The idea of a one-time treatment that could fix the genetic cause is exciting. But it is crucial to understand that these are still early-stage research projects. They are not available outside of clinical trials, and it could take many years before they become standard treatments if they are proven safe and effective. In summary, gene therapy and CRISPR gene editing show great promise for future LGMD treatments. Many studies have demonstrated the potential to correct genetic errors and improve muscle health in the lab. However, these approaches are not yet cures. More research is needed to ensure they are safe, practical, and effective for humans. For now, managing symptoms with supportive therapies remains the main approach. Anyone interested in experimental treatments should speak with their healthcare provider and consider participating in clinical trials. The muscular dystrophy community continues to push for research and hope, but it's essential to stay grounded in current scientific understanding and realistic expectations.